As mentioned before, each of the 4 nucleotides is labeled with a different colored phosphor and loaded. The small molecule real-time cell contains 150,000 wells called zero-mode waveguides, or ZMWs. A minimum of 40,000 ZMWs will contain readable templates and produce sequencing data. Each ZMW is visualized by a laser beamlet to record additions of fluorophore-tagged bases to a DNA template by a polymerase attached to the bottom of the ZMW. Each small molecule real-time cell is loaded with 1 sample in up to 12 SMRT cells. 12 small molecule real-time cells may be run within a 24-hour day. In step 4, a base is held in the detection volume and the light pulse is produced. As the DNA polymerase incorporates complementary nucleotides, each base is held within the detection volume for tens of milliseconds, orders of magnitude longer than the diffusion time. During this time, the polymerase-bound fluorophore emits a fluorescent light, the color of which corresponds to the base identity. Then, as a part of the incorporation cycle, the polymerase cleaves the bond connecting the nucleotide to the phosphor, allowing the phosphor to quickly diffuse out of the detection volume. In step 5, nucleotides held by the polymerase prior to incorporation emit an extended signal that identifies the bases being incorporated. This process of synthesis with phospholinked nucleotides takes place in a series of steps, as shown by the image below. First, the fluorescent phospholinked labeled nucleotides are introduced into the ZMW. Then the base being incorporated is held at the detection volume, producing a bright flash of light. After that, the phosphate chain is cleaved, releasing the attached dye molecule. And then the process repeats until the sequence is completed. Because of this particular way the label is attached, the DNA synthesized by the polymerase is completely natural and unmodified, leaving the polymerase without any memory of having processed an unnatural nucleotide. Inhibition of enzymatic activity, which is commonly observed by other types of labeled nucleotides, is thereby avoided, and the process is much faster and efficient. In step 6, the process occurs in parallel in up to thousands of ZMWs that make the small molecule real-time cell. The sensitivity of the ZMW-based single-molecule detection technology is 1,000 times greater than that of the existing microscopes. This enables it to discriminate signals against background noise while reading the individual bases of DNA as close as possible to the speed at which they are synthesized in nature. A prototype instrument enables recording of labeled nucleotide incorporations by single polymerase enzymes in real-time and high multiplex. In this highly parallel optic system, a detected flash of light is separated into a spatial array from which the identity of the incorporated base is determined. This is the works cited for this video. These are recommended videos in order to further understand how small molecule real-time sequencing works. In the next video, we will be covering how ion torrent sequencing, another type of next-generational sequencing, works.